This is the BioCentury Show.
Brought to you by BioCentury Grand Rounds. Join BioCentury this September in Amsterdam for the second Grand Rounds Europe, an R&D forum focused on translational science at the interface of industry and academia.
Welcome to the BioCentury Show. I'm speaking with Angela Genge today. She is a physician researcher who has spent more than 2 decades, I would say really at the center of clinical research for amyotrophic lateral sclerosis. So ALS, as it's more commonly referred to, that's a progressive, fatal, neurodegenerative disease that's been a really tough nut for drug developers to crack, but Angela remains optimistic, which I think will come through today. As a neurologist and a professor at McGill University, she's built one of the most active ALS trial sites probably in the world. Certainly, there have been few major sponsored programs that have come through Canada and not been in her clinic. An example is tofersen, right, which went on to become Qalsody. She was also involved in Radicava. So those are just 2 of the tiny handful of drugs that have actually received approvals for ALS. Scientifically, she came up in an era when multiple sclerosis was being transformed for the first time with effective treatments, and an experience that shaped her philosophy in ALS of, small wins can be meaningful. Take them where you can get them. Okay, one more thing. She is now also a chief medical officer at a biotech called ALS Pharma, which gives her yet another vantage point on the space. So, Angela, welcome to the show.
Thank you very much, Selina. Delighted to be here.
Yeah, I'm excited to have this conversation as well. It's such an interesting and an important indication with so much need. So maybe I will start with the challenges. Because, the word that I hear most often when people describe the challenges in ALS is heterogeneity. And that can mean different things. There's different types of heterogeneity, right? And I wanted to kind of march through them. But let's start first off with mechanistic diversity. So the biology behind the disease. That seems to be expanding rapidly, these different contributors. And what does that mean for drug developers?
So it certainly means, heterogeneity, as you said, Selina, is something that is used differently in different ways, but always seems to land on ALS when people describe it. And in some ways, I think they're bringing different things together. And it may not be as heterogeneous as people currently believe, but it is very true that science is leading us to look at a number of different pathways. And just when we feel that we've clearly answered one pathway as not important, another one shows up. So there's very much a whack-a-mole approach to mechanisms in ALS, and with historically the difficulty finding effective drugs, it's made that actually more intense. So if you look at the current pathways that we're actively exploring already in clinic, we have things related to TDP-43, including UNC13 and Stathman. We have inflammation that is currently being tackled. The current round is tackling things like TREM2 and certain other issues, pathways that are directly related to neuroinflammation. We also now have a focus on something called SARM1 that my colleagues worked on when they were doing their PhDs, and I won't tell you how many decades ago that was. And it's now becoming a target. So when you're an outsider or you're learning, all you hear is yet another pathway. The real question is, when it comes to developing therapeutics, are we getting lost in the forest, the trees for the forest? We do know right now that there's one target that in a subpopulation of patients, if you influence that target, you can be disease modifying. And that is SOD1. And, Qalsody is a perfect example of a development program that followed basic principles of drug development. The question we have is whether it's possible that there actually, even if there are many, many, many different pathways triggered, that at the end of the day, we will only need to treatment-wise target a handful. And what those handful of pathways are is really important. So we now have one pathway. That is the SOD1 pathway. If you have too much of it, if you develop misfolded, that's really the ALS Pharma story. And you clear misfolded, can you influence clinical outcomes? So we have one pathway proven by Koch with follow-on activity, not just ALS Pharma, but this program is the one heading into phase 3. Maybe there were only maybe 3 more. Maybe there are more, but I doubt there are going to be 25 pathways we have to target to get treatments. Even in the most intense oncology, you normally only have a few. At the end of the day, we're down to, several drugs, but not 25 for every disease. So
Yeah.
I think that's where we're going to end up.
Well, that's hopeful, right? It makes it more tractable. But let's talk about another aspect of heterogeneity, which is the rate of progression. So that varies dramatically from patient to patient. And I know that the field has struggled a little bit trying to stratify, fast progressors and slower ones, and just how to deal with that within the context of a clinical trial. So where are you at now? And do you feel like it's becoming a solved problem? What's the best practice?
Oh, that's a really, really good question. It's very current. I think we're really starting to land on the right approach. It's very true. It's a bell-shaped curve. There are people that die in less than a year, very rapid progressors, big problem with certain genetic forms. And there are those who live 10 years or more, and they contribute in other ways. The bulk is between 2 and 5 years, and what we call normal progressors are people that go from symptom onset to assisted ventilation, respiratory assistance, or death in 2 to 5 years. And that's probably the majority. The challenge when you're running a clinical trial, of course, is you want a homogeneous population in your treated arm and your placebo arm. A group out of Europe, and it's called TriCals, has developed a prediction model. It's actually developed a few years ago, but it's finally we finally have data sets demonstrating what they developed. And it is the TriCals prediction model uses between 12 and 13 different elements to determine at the point you're seeing the patient, say you're screening them to go into a trial, to determine what their progression will be. And it's scored. It has things as straightforward as site of onset or whether or not they have a genetic mutation. It also includes weight, because weight turns out to be one of the most objective predictors of prognosis. Change in weight from your symptom onset predicts the speed of progression probably better than any one single thing. It has a panel and it scores it, and then you can take the score. They've created this sort of system whereby you choose the range of results on the ALS prediction score for for patients that you want to include in your study. So if you want really fast progressors, you move towards one end of the scale. If you want slow progressors, the other. And if you want normal progressors, you go in the middle. We've tried other things in the past. We've tried predicting based on symptom onset to baseline and how many points in our functional rating scale was lost. That turned out to be not very good. Because people we're going on people's recollection of their symptom onset, and optimists see themselves as going slow and pessimists see themselves as going fast, and the reality is somewhere in between. We tried run-in so that you would baseline a patient and then do nothing for 3 months. That's basically intolerable for a patient and a clinician, and it also didn't hold up very well over time. So now we have this particular tool that seems to be working. It's been actually implemented in at least 4 sponsored trials in the last couple of years. Data is now out. They're now evaluating how closely the prediction model predicted the rate of progression based compared to how they really predicted in the trial. So that thing seems to be moving along.
That's great news. Out of curiosity, how does neurofilament fit into that picture? Is it part of that? Is it separate? Because I seem to recall that was an important piece, baseline neurofilament for CalCity.
It's a great question. The original prediction model was developed without neurofilament. Neurofilament has now been added, and it appears to make it even more robust, but it's not essential. You can do it without neurofilament. And one of the things about neurofilament is it's incredible. It was incredibly important for CalSODI, and it's probably very important for another drug that is different type of drug but works the same, knocks down total SOD1. But we now know from a couple of very diligent clinicians, the paper I'm most familiar with is out of Germany, that it doesn't have the same power in sporadic ALS. It's all over the map, and it can change quite quickly if you have a pneumonia, you have a fall, you have a head injury. So because it responds to the acute state of the patient, it bounces around a lot. So that's the biggest downside for neurofilament. It's not going to be as broadly applicable as we hoped.
That's really interesting. Yeah, thanks for that. Okay, so on trial design, marching through other challenges that have come up in my discussions over the years. That primary rating scale, ALS-FRS, has been criticized so many times. But I think you argue that actually the scale itself might not be the problem, but how it's executed. So you can talk a little bit about what does good execution look like, and how far has a field come in now being able to do that?
It's taken the effort of a group of people, and actually bringing the groups from the US, Europe, and Japan together. So what we have done is made a harmonized version of the training. So all of the questions get the same answers no matter where in the world you do it. So we've harmonized the training so that we've harmonized the scoring. We've identified that one of the most important variables is not the patient, but the rater. That a patient is actually quite consistent in how they rate themselves in the functional rating scale. But if you change the rater, the person asking the question and receiving the answers and documenting the score, a change in rater can change the score by 4 points. We currently believe that a change of 2 points between placebo and treated is clinically meaningful. And if you change a rater, just overnight, you can change the result by 4 points. So we're setting different rules. The training is now standardized. We're asking everyone to keep the same rater for the same patient for the duration of the double-blind period. And we're making things like that happen to really standardize it. It happened in MS, in the early 90s when I started by helping a colleague, a mentor of mine. I was actually a resident at the time. I started doing the EDSS for him in all the big early MS trials. And they had the advantage of the MRI, but they had to make a regimen around the EDSS in order to make it standardized, because it was the functional rating scale. It could not be something else. And the regulators insisted that it was part of the trials, which is where we are with the ALS-FRS. So we're fixing the training and the scoring. And I've just gotten word from a company just finished a trial. And in fact, the trial was negative. However, they were able to tell me that with this standardized process, it really decreased all the noise in the ALS-FRS. So we are now getting evidence that the hard work of a lot of people is actually paying off in terms of the functional rating scale, which is great news.
Oh, it's really encouraging.
Yeah, because we can't get rid of it. The regulators will not, until we have a fully vetted replacement, which takes years. it is our tool.
Okay, yeah. So, but when you think about maybe that longer term vision, do AI raters or continuous monitoring with digital trackers factor into a future where maybe you don't have to work so hard to train everybody and get consistency? Is there movement on that, or?
There is movement. I've actually used a few super interesting tools. They're not ready yet, and they're not ready for the regulators, but there's a very simple fix we're working on, and it's fascinating. The patient. If you do a self-report ALS-FRS, and you do that over time, it is more reliable, predictable, less variable than having a rater. Well, and we're just trying to come to terms together as a drug development and ALS community, because it's a real mix of industry and academia and clinical. But we looked at some data, and patients are more reliable. And when you think about it, Selina, it makes sense, because if the patient is an optimist, they're always going to score high. Even as they progress, they're going to rate themselves the highest possible. If they're a pessimist, they're going to consistently score themselves as low as possible. And it turns out that actually matters, that the self-report is actually quite robust. So we just have to get that into our drug development now.
Gotcha. Well, we have to go to a short break now, but when we come back, we're going to talk about biomarkers and the toolkit, how it's expanding, and ALS Pharma. So be right back.
This episode of the BioCentury Show is brought to you by the second BioCentury Grand Rounds Europe in Amsterdam, September 23rd to 25th. The second edition of BioCentury Grand Rounds Europe convenes the leaders shaping the future of biopharma. This time we meet in Amsterdam, September 23rd to 25th, gathering academic innovators, biotech CEOs, pharma R&D leaders and investors to debate the translational science that will define the next decade. Grand Rounds Europe focuses on what matters most: breakthrough biology, transformative technologies, and how to make early-stage R&D investable. Grand Rounds is where rigorous science meets real-world execution. Join BioCentury and regional host chairs Forbion and BGV and take a deep dive on translational opportunities in the Benelux region. BioCentury Grand Rounds Europe, where discovery moves toward impact. Register at biocenturygramrounds.com.
Welcome back to the BioCentury Show, where I am speaking with Angela Genge about ALS drug development and what can make clinical trials better. And so one of the things that needs to improve over the long term is the biomarker toolkit for understanding pathologies patients have, for tracking outcomes, responses to drugs. So can you just give us the overview, Angela, what's improved in the toolkit and where are important pieces still missing?
So I think what's improved is people's focus on it. So biomarkers turn out to be a really good thing in drug development, and particularly in ALS. ALS is not a field that's going to be conducive to fancy imaging because the patients can't lie flat or still for long enough. So we really had to focus on biomarkers. And the work that has led us to neurofilaments is rock solid. So we now have both serum neurofilament light and phosphorylated neurofilament heavy in the CSF. They have two slightly different sort of features. Because the PNFH is done in the CSF, it's done less frequently, but it's more stable longitudinally, and we do know what it looks like in natural history longitudinal studies. Because serum NFL is so easy to do, it's really been added to a lot of programs. Its drawback outside of the very specific SOD1 decrease type mechanisms. Its drawback is that it does vary with things that are unrelated to the disease. So you over time it works out, but there's noise in between. And it can take a while for it to change. What I find super duper exciting is we're actively looking for new biomarkers. And we're the big companies, the small companies, the academics, everyone is in this. And so I've recently seen, well, I've been involved in the development of microRNAs, that story around microRNA in ALS. And I've sort of watched and listened to the development of neuronally derived exosomes. And now we're in a point in time when we're looking at panels of proteins and metabolomics. But I think the most exciting part of this is actually who's in the game and what's in the game. So AI is in the game, guaranteed. But you also have some big pharma who've decided to invest in drug development in ALS, who are pumping their expertise in biomarkers brought in from oncology and immunology. And I think they're making big headway. So even in programs that I participate in, but I'm not as involved in as I am with ALS Pharma right now, I see this really important energy going into it. I can tell you the regulators expect things to be developed properly. They have certain standards. Whether you talk to CPATH or directly with the FDA, you will get clear guidance on what you have to do. But I've probably seen 8 or 10 efforts in the last year and a half. So I'm very hopeful of this of biomarkers being able to give us signal prior to clinical improvement that we're on the right path, because that's what they're being used for, right? It's a surrogate of clinical benefit. It doesn't replace clinical benefit, but it gives you heads up that you're on the right path.
Right. So neurofilament is used in that way. But when I think about, people will often say, what we really need is a TDP-43 marker of some kind, PET or otherwise. What would that do for the field?
Right. I think that will be huge, and I can tell you several groups. The academic group in Florida is very involved in this. One of the big pharmas is very, very involved in this, and then there's small sort of biomarker startups are also. So I think we're not that far from a biomarker in TDP-43. I've seen a couple, so I think it may be in the next 2 years, actually. I think it's finally moving that quickly. If we're unlucky, it'll take 5. It's not going to take 10. Too much brain power, too much technology is being applied for it to take that long.
Okay. And then what about SOD1? Because that is what ALS Pharma is working on.
Right. So SOD1, we have an assay for normal SOD1. What ALS Pharma developed was a Simoa assay looking for misfolded SOD1. So SOD1 is an important protein in the body. We can't live without it, right? It's 1% of the CNS. When it misfolds is when it causes problems. It's a toxic gain of function pathway. You misfold, it causes motor neurons to basically malfunction and then die. It can be, but what has been missing or elusive is an assay to test it during life. There's a lot of activity and a lot of evidence in a lot of labs that and postmortem specimens across the spectrum of ALS patients, you can document misfolded SOD1. What ALS Pharma did was developed an assay that actually measures it in CSF. So you're able to see in not only SOD1 patients, but also in sporadic patients. There are sporadic patients who also have elevated misfolded SOD1. So the concept is, although we understand the pathway through the SOD1 patients, that it's not exclusive to people with the mutation, that wild type, which is normal SOD1, can also start misfolding and start move along like the expression people use is a prion-like manner because it has this spreading phenomenon. You misfold in one region and it starts to trigger more misfolding and more misfolding.
Yes, that is a common theme across neurodegeneration. We see it with tau and Alzheimer's. We see it
Yes.
with alpha-synuclein and Parkinson's disease. And kind of one of the big debates has been like, okay, well, those proteins like SOD1, their main functions or their main activities or main home really is intracellular, but then they do this prion spreading thing, which is extracellular. So do you have to target both of those? Will one work? And so far, this intercepting the prion-like spread for the other targets, the tau and synuclein, it has yet to pan out. It might still. But do you think there's something that would make SOD1, I don't know, different or more amenable or
So that's what we're seeing in our phase 2 study, that we target misfolded SOD1. You get it out of circulation. It's an IV medication, so it's also has the opportunity for retrograde transport. And then if you clear the really active forms which are circulating, we have evidence both with biomarkers and with clinical that you can actually change the course of the disease. So it's a small phase 2. That's why we need the large phase 3, but it's pretty exciting. And the fact that we've seen this misfolded SOD1, and we've basically confirmed what autopsy programs showed. We see the misfolded SOD1 in sporadic patients in life. So the target, it may be a treatment that works in patients with non-genetic forms of ALS.
Yeah, it's really neat. You could say this is some of the first phase 2 data from any of these neurodegenerative diseases, suggesting that this intercepting of the misfolded seeds extracellularly might be therapeutically beneficial, which is a big theme. Yeah. Okay.
Yeah.
So there's TDP pathology, there's SOD1 pathology, which initially I kind of thought was only relevant to those mutation carriers. So how are you getting a sense of like who among the sporadic population this is going to be most useful for, and how big do you think that population might be?
So it's interesting because we actually, until we get a biomarker, we assume that most patients have most sporadics are driven by TDP-43 because of pathology, right? Because there's autopsy reports. But the evidence we're having so far is that at least a third of sporadic patients potentially respond to the therapy, at least a third, like SOD1 patients. And figuring out just how many, what proportion, is a big deal. And then the other question, which you alluded to, is it an either or situation, or is it possible that somebody has TDP-43 pathology and still has some motor neuron loss related to misfolded SOD1? Based on a very few case reports, both in autopsy and with one or two of the patients in our program, which were there because they didn't have SOD1 and ended up having other mutations, that it's not an either or. You can have a mutation or TDP-43 pathology and still have misfolded SOD1. So it could very well be that we are down to, I'm not saying there's only two, because I think we're going to probably have 3 or 4 key pathways that when you target them well, you could change the course of this disease. But I don't think there's 100. I'm happy that we're seeing 100, but I think it will whittle it down, and I think there will be a handful. And right now, the evidence in this program is you've got the SOD1 patients, the patients with the mutation, and you have a proportion of patients with sporadic disease, meaning they don't have the SOD1 mutation, that also have the misfolded SOD1 circulating, and you can target it and have an effect. So that's big.
Yeah. So right now, I presumably what you would like to study, if you could study, is how SOD1 and TDP-3 interact, how they both interact in the presence of the drug, which you presumably can't do now as you're designing your phase 3 trial.
Right.
But what what can you design into the trial, given everything that you've said in this discussion about optimizing for success with today's challenges and tools?
Yeah.
What are you thinking?
Well, we're definitely looking at making sure that all the arms of the trial are balanced in terms of levels of misfolded SOD1, so we get a clear picture at the end of the trial. And we're also going to make sure we're balanced with somehow with NFL. But I think what we're going to really do is look at some of the traditional, well-studied outcome measures to see just how much what we see assay-wise with biomarkers actually translate into clinical effect. I think that's what we have to do. We have evidence that it does, but now we need it in larger numbers. What we're happy about is this is an easy to give drug. So we're not going to have to worry about an IV, any reaction. We will be looking at developing different kinds of biomarkers related to misfolded SOD1, but what will be integrated and what will be done at the end of the study, we'll see. And I feel TDP-43 is also really important. So I don't see this as as one or the other. I think this is the two main paths. I think we need to explore both of them, particularly for patients with sporadic disease where we can't blame a gene.
Totally. And if I remember correctly, among the lessons when we were talking earlier that you said came out of the tofersen trial and other trials is really just how long you need to wait to see changes on that rating scale. So you want to talk a little bit about that?
I think I'm glad you brought it up. So we know very well what tofersen has done. We know the timing on the biomarker and we know the timing of the clinical benefit. If you target it differently, and so a target that's circulating misfolded SOD1 is a different target. We don't touch normal SOD1 with this. What we see is a different timing of the changes in neurofilament and actually a different timing of the clinical effect. So the clinical effect occurs a little earlier, the biomarker effect occurs a little later. Everything is related to the mechanism of action, Selina, right? So if you're targeting normal levels of something that have a downstream effect, you have to knock it down and then see the clinical effect. And so we're looking at doing the double-blind trial for 12 months so that we give adequate time to understand. And I see this as the trend across the field. I think the experiment that was doing 6 months double-blind, and there's still debate, but I think that was an experiment, and I understand from the patient perspective why. But if we miss a target, because we nearly didn't get tofersen because of it, we're actually doing more harm than good. So it's better to take the longer period to do the double blind. There's debate, 9 months, 12 months, 18 months. Very Canadian, straight in the middle, 12 months double blind, and give yourself a chance to be able to demonstrate efficacy so you can go quickly to approval.
Yeah. So we've talked about a lot of lessons that have been learned. Have there been lessons that you think people should have learned, but they haven't fully absorbed yet?
Boy, interesting way to say it. So I think we have to be rigorous in our trial design. I think we have to use our phase 2 trials, and anyone out there involved in drug development in ALS needs to know phase 1 is safety and tolerability. You need to really be diligent in your phase 2 trial. If you want to test something, test it in that phase 2. Add in your special AI-driven data entry. Use both your validated and your more experimental biomarkers. I've learned from the field of FSHD, facioscapulohumeral dystrophy, that had absolutely nothing. And then a couple great biotechs came along They did really innovative things. I had one company that was injecting the muscles directly. Then we had another company that was doing muscle biopsies every 3 months and found out the patient tolerated it. And now we're on the verge of actually treatments that really are disease modifying in the most common muscular dystrophy in adults. And all of these programs had these phase 2 trials with lots and lots and lots of outcome measures, and that shaped the phase 3 because they were able to look and choose what was best for following the patients, and they've done it in 15 years. 15 years is not very long to go from first drug ever exposed to this population to something that's in the brink of being approved. That's very fast in drug development. In neurology, I live in neurology.
No, indeed.
And I grew up in the era of HIV/AIDS, and I saw what the power of a community working together could do. Took care of the young men who were dying in the 80s with no hope, and worked in it actually simultaneously with MS until the triple therapy was developed. If we work together and do all this stuff well, we get to the answer.
Yeah, and that's one thing you definitely have in ALS, a very active, involved patient community that can help you recruit for trials and things. But I think we are out of time for today. That was really wonderful, illuminating, and thank you for your perspectives.
Thank you for taking the time, Selina. It was great.
Until next time. Bye.
Brought to you by BioCentury Grand Rounds. Join BioCentury this September in Amsterdam for the second Grand Rounds Europe, an R&D forum focused on translational science at the interface of industry and academia.
Kendall Square Orchestra provides the music for the BioCentury Show. The group connects science and technology professionals and other members of the greater Boston community to collaborate, innovate, and inspire through music while supporting causes related to healthcare and education.